Biological Sciences here at the University of Aberdeen is full of researchers studying the impact of climate change on organisms, from tiny microbes to huge whales. We have a vibrant community of over 300 researchers here in the School of Biological Sciences, and they're split into 3 themes or programs, and that is ecology and evolution, environmental physiology, and biological interactions in soils. One key area of research, though, is experimental aquatic biology. And what is that? Well, quite simply, it's just doing experiments on organisms that live in water, and that can be marine water, so seawater, or freshwater like our rivers and lochs. And why is that important? Well, our planet is covered by 70% of water. We are a blue planet, and the organisms that live in our oceans and in our waters are crucially important to us as a food source, for example, also as a source of biologically active compounds. For example, many of our medicines come from animals and organisms that live in water. In order to carry out aquatic experimental biology, we need aquarium facilities to keep our animals alive. Luckily, here in the School of Biological Sciences, we've got a state-of-the-art aquarium facility where we house both our fish and our shellfish.
I'm Professor Sam Martin, the Director of the Scottish Fish Immunology Research Centre here within the School of Biological Sciences. Our research focuses on many aspects of fish health and fish nutrition. The aquaculture industry is the fastest growing food sector, but it is constrained at the moment by infectious disease and fish welfare. So behind me in the aquarium we keep rainbow trout and Atlantic salmon. The water is of very high quality and it's recirculated, and we monitor continually oxygen levels, carbon dioxide and ammonia. We have automatic feeders on our tanks to ensure that all tanks get equal amounts of food to be able to determine food consumption. Another main aspect of our work is developing new diets for aquaculture, where we have moved away from marine-sourced nutrients to using terrestrial plant proteins and oils, which are much more environmentally friendly. So as well as the work we do down in the aquarium, a lot of our research gets done in the research labs in the School of Biological Sciences. Much of our work within the labs involves looking at cells, which have either been extracted from the fish or we have immortalised cell lines. This gives us the ability to understand the fundamental workings of the fish immune system. We also do aspects of developing new vaccines for fish as well, and much of our work relies on cell culture to understand how the immune system actually responds to the vaccines and how these can develop protection against viral and bacterial pathogens. This is a really exciting time for working on fish, because the whole genome of Atlantic salmon and rainbow trout have recently been sequenced. So we are using a whole repertoire of modern genomics approaches for our research, for both aquaculture and fundamental fish immunology research.
Research in my lab aims to understand how shellfish build their shells, and this has many impacts. One of the main ones is in understanding the consequences of climate change. As the ocean becomes warmer and more acidic, we're worried about the fate of these calcified hard structures. We also believe that shellfish could be a really sustainable source of protein for the growing human population, especially when grown in an aquaculture context. So research in my lab uses the aquarium facility to house our adult broodstock, and that means that we can get embryos from these animals any day of the year. We then bring those embryos up here to the research laboratory. In the research lab, we carry out experiments. We manipulate those eggs, we try to do something called gene editing, and we study the function of genes through developmental time. We also study processes such as aging and regeneration of the shell. One of the key aspects of the research we do is to study the developmental stages of building a shell. So we want to understand, as the animal is putting itself together in those early developmental stages. And to do that, we have to use a combination of facilities and techniques. We do microscopy, molecular biology, and something called paraffin histology here in the laboratory. One of the really exciting things at the moment for experimental aquatic biology is the advent of a new technology called CRISPR-Cas9 gene editing. And this means that we can go into the animal and alter the genes and see what happens. This helps us to understand how those genes function inside of the animal. I feel really lucky to do the job I do here in Aberdeen. Every day we get to study the natural world and uncover the secrets of Mother Nature. So I hope you've enjoyed learning a little bit more about what we do here at the University of Aberdeen.